Right, so we are now looking at enzymes. We've already done those cell specializations, and please, that's very important. Make sure that you know on cells, specialized cells. What are the adaptations and functions? That is very, very important. Okay. Now, enzymes. We define an enzyme as a biological catalyst. A catalyst is just any molecule that will speed up the rate of a chemical reaction. But the important thing is that when this catalyst is speeding up the rate of a chemical reaction, it is not going to be changed. Okay? Let's say, for example, you are reacting, let's give an example, you are reacting two things. One of them is going to be an enzyme. Say you have got this structure. This is an enzyme, and then we are reacting it with this compound. So this compound we call it as a substrate. Okay, substrate. And then this one is the enzyme. So what happens that the substrate fits on this part of the enzyme. So when that happens, what you'll be able to see is now going to be something like this. Here is your enzyme, and then the substrate is going to be able to fit here. Okay. So what we have formed here is known as the enzyme substrate complex. That is what we have formed. At the end of the reaction, the enzyme is going to convert this substrate into the product. The enzyme will come out just the same way it is. It will come out like that. But then the substrate, you are going to see it is now going to be broken down. So they are going to be different pieces. It's no longer called the substrate. It's now, this is still the enzyme, but this one is no longer called the substrate. It's called the product. So very important, enzymes will not change after a chemical reaction. Why? Because they are catalysts. A catalyst is a molecule that will speed up the rate of a reaction while itself is not changed. Now the question should be, how does an enzyme speed up the rate of a chemical reaction? So there is something which is known as the activation energy. Very important. For a chemical reaction to happen, imagine if you want to form water, what are you supposed to react? Very good. You need oxygen and hydrogen. So if you have got hydrogen and oxygen, they will not just form water because hydrogen is there in the atmosphere, oxygen is also there in the atmosphere, but they don't form water. Why? Because there is a certain amount of energy that they require for them to form water. Okay, so this amount of energy, this one, from the beginning where you have got the reactants here to the highest point, that is known as activation energy. Activation energy. And then here now you're going to have your product, which is water. So we have got what we call activation energy barrier. So this hill that you can see is too long. So you need a lot of energy for you to be able to reach the top. When you reach the top, that's when you can now form water. Otherwise, as long as you've not reached the top, you will not form water. Let's say maybe at this level we have written R there is 20 joules. So maybe let's say at this level on top it is let's say 100 joules. So you need to reach all the way up to that top. So you need to add energy. But what an enzyme is going to do is that it is going to come and lower the activation energy barrier like this. You can still form water, but look, the Kama mountain that is there is small. You can easily climb this mountain. you reach the top. Immediately you reach the top, then you can form water. Okay. So an enzyme lowers the activation energy barrier, and that is how it will be able to speed up the rate of a chemical reaction. Is that okay? Is this making sense, or there's a question? Okay. Another thing that you need to know about an enzyme is that an enzyme is going to have some sites where things can attach. Here is our enzyme. Let me even shade it. So this part where the substrate attaches is known as the active site. And then there are some other places which are not active sites where other molecules are able to attach. Okay? Other molecules. For example, this space which is here is not the active site. So it's known as the allosteric site. Now, mostly enzymes do not work because for them to work, they need what we call a coenzyme. Co means together. So there are some molecules which work together with an enzyme, okay, for the enzyme to be able to work. For example, say you want to lift a very big block. Of course, you can lift it, but alone you might not do it effectively. So you need another person to help you hold the other side. Okay, so that is what coenzymes do. They'll be able to attach to the allosteric site of the enzyme to activate that enzyme. There are some enzymes that work inside the cells. We call them intracellular enzymes. Other enzymes work outside the cells. They are called extracellular enzymes. Okay, so that is it. Please don't forget that an enzyme will be able to react with a substrate, and then you'll be able to form the enzyme substrate complex. And then now the enzyme substrate complex will dissociate. Now you are going to form the enzyme and the product. The substrate has now been converted into product. You need to know the characteristics of enzymes. Very, very important. It can come in bio paper 2 or paper 1. So you need to make sure that you know. The first thing is enzymes are proteins in nature. Most of them are going to be proteins. What makes up proteins? Do you know what makes up proteins? The basic unit? Mm. So, proteins are made up of amino acids, meaning that since enzymes are mostly proteins, they're also going to be made up of amino acids. Another characteristic is that they are catalysts, meaning they speed up the rate of a reaction while themselves are not changed. Another characteristic is they catalyze both forward and reverse reactions. So just what I wrote here. We can have an enzyme reacting with the substrate to form enzyme substrate complex, and then we form the product. We can as well get this product react to the enzyme and then come and form back our substrate. So it can either go forward or backward. That is why you can see we are writing the arrows like this, forward and back. Okay. Another thing is that enzymes are specific, just the way a lock and a key is. Okay. Each key can only open one lock. So that is the meaning of specificity. Enzymes, each enzyme can only work on one substrate. For example, if this enzyme is working on glucose, it can't work on amino acids. That's the meaning. Again, there are a lot of another property of enzymes or characteristic. There are a lot of factors that can be able to affect them. Factors like temperature, pH, substrate concentration, enzyme concentration, and also inhibitors and cofactors. You need to make sure that you know all of this and how they do it. Okay, make sure that you know. The first thing is, let's explain what do we mean by enzymes are specific. We can use the lock and key to explain. So this is the enzyme, this is the substrate. The substrate will fit exactly in the enzyme. That forms what we call the enzyme substrate complex. So just like a key fits into the lock, the enzyme also fits into, the substrate also fits into the enzyme. That's the lock and key. Now let's talk about the different factors that affect the rate of a chemical reaction. The first one is temperature. Okay, how does, not really the rate of a chemical reaction, but enzyme catalyze the reaction? How does temperature affect the rate at which enzymes do their function? So each enzyme has got a temperature at which they work very well, and that temperature is known as the optimum temperature. Below that temperature and above that temperature, the enzyme is destroyed. Okay, like for example, our body is mostly the temperature in our body is going to be 37 degrees. So enzymes which are in our bodies work well at at the 7 degrees Celsius. Any temperature below 37 degrees Celsius, these enzymes will not be able to work well. Why? Because the enzymes are inactive. Low temperature inactivates the enzyme. But then for temperatures which are greater than 37, for example, you get 40, 45, the enzymes are going to be denatured. Remember we said that enzymes are mostly proteins. So proteins have got a way the amino acids have to be arranged. So if you increase temperature, it's going to destroy the arrangement of the amino acids, right? And so that is what we call denaturing. How about temperature? Okay, how about pH? We've talked about temperature. Now, enzymes the same, they work well at a specific pH. Yes, please. Okay. When and each enzyme has got a specific temperature at which it works well. For example, our body, normally our body temperature is 37 degrees Celsius. So enzymes in our bodies, meaning that they work well at this temperature. That temperature is known as the optimum. It's a maximum temperature at which the enzymes will be able to work well. Below 37 degrees, the enzymes are going to be inactive because the temperature is low. But if you increase the temperature very much, you start to destroy the enzymes. So that destroying the enzymes is what we call to denature. So above the optimum temperature, enzymes are denatured. Below, they're inactive. That's why they will not be able to work well. Is that okay? Okay. Now for pH, pH is a measure of how acidic or alkaline a substance is. For example, hydrogen, lemons. If you eat lemons, you feel the way they feel. Lemons are acidic, and then soda is basic. What determines the acidity of a molecule is hydrogen. I saw this question in one exam paper. So that was chemistry. The determinant of acidity is hydrogen. Okay, that's what determines the acidity of a molecule. So to measure the acidity of a molecule or the basicity, in other words, to measure the pH of a molecule, we can use a pH scale. A pH scale starts from 0 all the way up to 14. A molecule that is not basic and not acidic is a neutral molecule, and it has got a pH of 7, which is the middle of the scale. When you start to go up, the molecule is becoming more alkaline. Alkaline is the same as base. Okay, so if you have a molecule that has got a pH of 14 and a molecule that has got a pH of 11, the one which has got a pH of 14 is a stronger base, or it is more alkaline, while the one which has got a pH of 11 is a stronger acid, because when you are moving in this direction, the strength of the acid is becoming more. Now, when you get to be below 7, so from 0 to 6.9, that pH is acidic. From 7.1 to 14, that pH is basic. So acidity is going to increase as you go in this direction. Basicity increases as you go in this direction. So a pH of 1 and a pH of 4, which one do you think is going to be a stronger acid? Between between a molecule with a pH of 1 and one with a pH of 4. Very good. pH of 1. So what happens that molecules again have got a certain pH at which they work well. That is known as the optimum pH. So below the optimum pH and above the optimum pH, the enzymes will not be able to work well. All right? The enzyme will not be able to work well because they are going to be actually destroyed. If an enzyme works well in a pH that is acidic, you make the pH to be basic, that enzyme will be destroyed. So that is what pH does. And then now the enzyme concentration. What is enzyme concentration? Enzyme concentration is how many enzymes do you have. Now say we have got 20 molecules or 20 substrates molecules are supposed to work on. We only have one enzyme. So what will be happening is that this reaction is going to be slow. Why? One enzyme will first take up one molecule and then convert it to product. After it is done, it now gets another molecule, converts it to the product. After it's done, just like that. So it's going to be slow. When you start to increase the concentration of enzymes, let's say you make now the enzymes to be 2, to be 3, to be 4, it will be very easy because imagine you are doing work and then you are only alone. You will not be able to do it very well. Okay? So when you increase the enzyme concentration, the reaction will becoming much faster. You'll be doing work more and more faster. But then there will be a point that is where even when you increase the amount of enzymes, the reaction will no longer change. Because if you have got 20 substrates, the fastest reaction is going to happen when you have got 20 enzymes. Why? Because each substrate is going to have one enzyme. Okay? But then let's say we take the substrates to 40. This means that these 40 enzymes, only 20 will get these 20 substrates. The other 20 will not be doing anything. So increasing the concentration of the enzymes will not change the rate of the reaction because you don't have what those enzymes are going to be able to work on or act on. You don't have anything. Is that okay? Okay, so that is the same that happens with substrate concentration. Now it's just that here you have got substrates. Let's say you have got 20 enzymes, but then they don't have what to work on. You only have one substrate. When you start to increase the concentration of the substrate, the rate of the reaction will be increasing. Let's say if you have 10, 20 substrates, it will be able to fit 20 substrates to work with the 20 enzyme. But if you increase the substrate to 40, it means that 20 of the substrates will be taken up by the 20 enzymes. The other 20 substrates will have nowhere to go. So the rate of the reaction will no longer increase. Okay. Even when you increase the concentration of the substrate. So that is also the substrate concentration. We also have what we call inhibitors. An inhibitor is a molecule or a substance that will be able to slow down the enzyme activity. Now, inhibitors can do their function in two ways, mostly, or three ways, let's say. You can have competitive inhibitors, non-competitive inhibitors, or anti-competitive inhibitors. So what happens is, you know that for an enzyme, you have got the active site. This active site, what is supposed to bind there is a substrate. In a competitive inhibition, instead of the substrate coming to bind here, an inhibitor will come and bind here. So meaning that this enzyme will not be able to convert the molecule it was supposed to convert into product, because the place where the substrate was supposed to bind, there is something already binding. Think of it like there is a guy who likes you, but because you already know, okay, this girl has a boyfriend, that boy is obstructing that other guy from approaching you. So that is how competitive inhibition works. The active site contains some other thing. So that prevents the substrate which is supposed to bind to to the active site from binding there. Non-competitive inhibition, how does it work? Remember we said that for enzymes they also have what we call the allosteric site. So say this molecule, this enzyme is supposed to be like this. The substrate is supposed to look something like this so that it can fit in here. Now in an competitive inhibition, what happens is that an inhibitor will come and bind to the allosteric site, not to the active site. When it binds to the allosteric site, what happens is that the shape of the active site changes. Look at how the shape of the active site has become. So because the shape of the active site has changed, the substrate can't fit to this lock. It can't fit. This key can no longer fit to that lock because you have destroyed the lock. So that is how a non-competitive inhibitor is going to be able to work. Is that okay? Okay. And then for uncompetitive, please, it's different non-competitive and uncompetitive. For uncompetitive, you are going to have your enzyme, and then the substrate is already attached there. So this one we call it as the enzyme substrate complex. So the inhibitor will come and bind, okay, to the enzyme substrate complex, and then that will be able to distort this molecule such that the enzyme will no longer be able to convert the substrate to the product. So that is what happens. Examples of competitive inhibitions. Let's talk about non-competitive inhibition. Is a poison. So poisons can actually get to act on the non-active site of the enzyme, and then they will not be able to work. We also have what we call cofactors. Cofactors are just opposites of inhibitors. For an enzyme to work, it needs a molecule. Okay, so we have got what we call coenzymes. Now, coenzymes, these are molecules that will help the enzyme to work. So without coenzymes, enzymes can't work. It's like you have got a gun, but there is no gunpowder, so they can't work. Coenzymes, these are going to be made up of mostly amino acids, okay, things like that. They are actually organic compounds which will be able to activate an enzyme for it to be able to work. Now, coenzymes, which are inorganic. Inorganic meaning that they are not made up of carbon. Those are what we call cofactors. So cofactors are simply coenzymes which are inorganic. The examples of this will include just metal ions like iron 2 plus, zinc 2 plus, metal ions. But in organic molecules which are coenzymes will include things like uh, what's this? The word has gone. Minerals, okay, like the mineral, not minerals. These guys here that you can see are minerals. So we have got vitamins. So that's what I wanted. The vitamins. These are organic because they are going to be made up of carbon, hydrogen. So molecules made up of carbon, hydrogen are actually organic molecules. Okay. So this is on the cofactors. Is there any question on here? Okay. Now, how do you get to name enzymes? You name enzymes depending on the molecule where they're acting on. If the molecule is acting on a starch, now starch is made up of two things, amylose and amylopectin. So just get the amylose. We'll remove the last three letters of amylose, OSE, and substitute it with ASE. Enzymes will always end in ASE. So it becomes amylase. And then for maltose, just remove the last three letters and substitute with ASE. Becomes maltase. Sucrose, sucrase. Lactose, lactase. Peptide, peptide. Now for this one, just remove the last T, E, and then put ASE. Peptidase. And then lipids, just remove the S at the end. Actually, for this one, remove the last three letters, lipid, and then put ASE, that becomes lipase. Okay. You can have some other enzymes that do not end in ASE. These ones are special. They can actually end in IN. For example, pepsin, trypsin, renin, they are ending in IN. That is how you can get to also consider those. Now, what are the uses of enzymes? Enzymes are going to have different uses, and one of the uses they can actually be used in the industry. And in the industry, some enzyme can be used to make detergents, making things like soaps. Okay. Others can be used in brewing and baking, making beer and also in baking. For example, I've got the enzyme zymes. This one is found in yeast. Okay. So when you are baking, you are going to mix flour, water, sugar, and yeast. And that to make a dough that for it to be able to come out like that to become big. So yeast will be able to secrete the enzyme zymes, and this enzyme will be able to break down sugars which you are mixing, and you are going to be able to form alcohol and carbon dioxide. This carbon dioxide which is produced is what forms bubbles which make that dough. Okay, that's how the flour starts to appear to be a lot. When you are brewing, like making the beer and other things, cereal seeds are going to be soaked until they start germinating. If you have ever seen where they make cachaso, okay, you are going to get maize, you soak it in water, you put it in a plastic until it starts to germinate. And then during this process of germination, the starch is going to be broken down to maltose by the enzyme, amylase. That is because the starch, remember we said that it's made up of amylase and amylopectin. And that amylase and amylopectin is actually made up of maltose, a lot of maltose molecules. And a lot of maltose molecules are made up of actually glucose. So if you put two glucose molecules together, they form maltose. If you put a lot of glucose molecules together, they'll now form either amylopectin or amylose, which forms the starch. So these enzymes can also be used as sweeteners and also in getting to make drinks like that. Can be used as sweeteners. Enzymes can also be used in the drying industry. Okay, in the dairy industry. Dairy industry is the industry that has to do with the production of milk. So when you want to make milk, you want to make from milk things like yogurt, cheese. You need enzymes to be able to do that. So that is the dairy industry. You can as well use enzymes in tanning of leather. Tanning is just getting to make leather soft and pliable. Pliable means that you can actually extend it, like your skin. If you pull your skin, it comes back like that. That's tanning, or that's pliability, pliable. So trypsin is utilized to digest proteins in the leather during tanning. So for you to be able to make these molecules soft, you need the enzyme trypsin, which is important in tanning. Okay. Enzymes are also going to be used in extraction and processing of fruit juice. For example, cellulase and pectinases. These are going to be used to increase the juice yield and prevent gelling of the juice. Now, cellulase means it works on cellulose. Cellulose is found in the cell membrane of plants. Also used in the tenderizing of meat. Okay, so you want to make the meat tender, so you can use the enzyme trypsin to be able to pre-digest some foods, like for example preparing baby foods, to digest them in advance so that they will not be that strong, that is to make them tender. Okay, so those are functions of enzymes. In the next class we'll talk about the next topic, or we'll not do classification of organisms because this one is supposed to do it much later, but we'll do the next topic.